Okay. So thank you everyone for attending today. So before we introduce the speaker, so I realized that there's something that I've been missing out on introducing on an important aspect. So Panoramics offers events under our Panoramic chapters. So this chapter comprises of the Enlighten Me, the Challenge Me, and the Inspire Me series. So today is our first event from our Challenge Me series. So this series introduces seminars and tutorials and other events that is meant to challenge moving beyond conventional thought or assumptions. So it includes topics we take for granted, and perhaps normalization is one of them. So today we have invited Dr. Rhonda Baker to deliver us a lecture on an examination of the evolving views on normalization for single-cell RNA-seq data. So Rhonda is an assistant professor of the Department of Statistics at the University of Florida, and I will post a link to her website on chat later. Please feel free to post your questions on the chat section. And I'm now going to hand it over to Rhonda to challenge us. So the screen is yours, Rhonda. You can take it away.
Thank you. All right, I'll try to keep the chat open if anyone has questions, but it's really easy to lose track of it. So if anyone wants to stop me, I think the best way it would be for Shamini to interrupt me and say there's like a critical question, but I'm happy to stop anytime. It's just hard to see everything with the screen. Thank you for the great introduction. Oops. As mentioned, I'm going to talk about normalization. I usually have an introduction of what single cell is, and I felt weird not even having a slide, but this is the only thing I have because I'm assuming everyone here already knows about single cell RNA-seq, and you had yesterday you had another talk going through all the basic analysis steps. So as long as nobody's lost by this slide, I'm gonna move on. All right, so let's talk about normalization. So one thing that is, I think most people do realize when they do single cell analysis is there are these pre-processing steps, and normalization is one that's mentioned in every kind of pipeline that's out there. And it's done before any type of downstream analysis, whether it's differential expression or looking for trajectories or even doing cell type identification. So there's some already insinuation that it affects these in some way. And so we're going to talk about methods for normalizing later on and how maybe it affects those different downstream analyses in specific ways. But just to get all on the same page, we're going to do a refresher on what normalization is in the very simplest sense. So not even thinking about single cells yet, but just what is normalization trying to address, underlying that definition. So I have an illustrative example here. And so we're going to pretend that sequencing is like drawing blocks from a bucket. This is a more modern version of the marbles from an urn that statisticians love using. So it's a little bit more updated. So here's our bucket. Let's assume that the different colors represent different genes and that the buckets represent samples. And so here I have one sample. And then let's also assume that just like with sequencing, where we amplify things a number of times, that the number of blocks here is infinite, right? So like imagine you have a big box of Legos, and if I wanted to take 1 million, I could keep drawing. So even though I have 40, 10, and 50 here, it's more of just a proportional representation of what's in the bucket rather than the absolute value. Okay, so let's start with the very simplest scenario where our two samples or two buckets are pretty much identical, right? So they're proportionally, the blue is the same, the red and the purple are exactly the same. And so we can represent the ground truth here just using proportions to the total number of blocks or gene counts. And let's say that I'm going to do a simple sampling procedure. So I could go into R and I could say, hey, I have these probabilities, and let's say I want to draw 96 blocks with these probabilities. And then for sample 2, I want a total of 39 sampled with this probability on the right here. And so let's just say one single sampling that I performed, these are the values that I get. So the totals are essentially random, but you can see that sample 1 was sequenced almost twice, 2.5 times more than sample 2. And because of that, in the sample 1, every gene looks like it's higher expressed. And so if you just were to not perform any type of normalization or accounting for these differences in total sampling or sequencing, you can immediately see why there's potentially issues. And so we'll once I introduce different types of methods, we'll see how we actually do normalize these counts to get more consistent comparisons across samples. Okay, so let's do something a little bit more complicated. So in this case, we have a differentially expressed gene in the purple block, so from 60 to 500, but everything else is exactly the same. So again, just assuming I can represent this ground truth as proportions and then perform some type of sampling procedure. Again, these totals are pretty random, but in this case, let's say sample 1 was sequenced almost twice as much as sample 2. And again, we see the same effect where most of the genes or blocks are higher in sample 1, with the exception of the purple block. So in this case, this truly differentially expressed gene actually appears to be equal. And so again, this kind of underlies the need of, we need some type of normalization to make the counts comparable across samples. And then example C here is just another variation on this. So instead of being differentially expressed in the sense of overexpression, in this case, bucket 2 has genes that are just not expressed for some reason. And it has some in common, but there's more that are actually just off and equal to 0. And so again, representing these proportionally, we can perform just a sampling. In this case, sample 1 also got sequenced more, just again by chance. And so when this happens, now of course the ones that are compared for 0 in the purple and the green and in the yellow, we do see that there is the differential expression of the non-zero values to 0, but the blue and the red here are supposed to be equivalently expressed, but here we see an increase in expression of sample 2, which we know is not accurate. So again, very similar to the sequencing situation where we do get differences in sequencing depth per sample. And so when a biologist goes to perform an experiment, you do try to put equal amount of samples on the plate. If you're multiplexing, you try to make it the same. There's robots now that try to help you do this, but it's still hard to be exact. And so for many reasons, we actually do see this variability in total amount of samplings when we're sequencing. And so normalization methods will typically adjust for differences in the sequencing depth by calculating some type of global scale factor. So it's applied to each sample across all genes. The most popular methods that you've probably seen are the counts per million, or for single cell, the counts per 10,000, which is very common. And if you worked with bulk RNA-seq, you would have seen TPM or RPKM, which are very similar, but they need to do a length adjustment. And so what we do is we typically will divide each sample by its total count and then multiply by this constant, whether it's 1 million or 10,000. Okay, so seeing this really simple example A, going back to example A, if we just apply this, counts per million, in this case, I'm going to use counts per 100 because this is just, toy numbers. And so essentially I'm just dividing each value. So 42 divided by 96 times 100, and doing that for every cell here. Now we can see that between samples, our counts are equivalently expressed. So we've essentially removed this effect of sequencing depth or sampling, and things look great. In example B, remember we have this differentially expressed gene here in purple. And so if we apply this counts per 100 normalization, so now I've fixed the totals to be 100, but when we look at the comparisons for each gene across samples, there's some really obvious problems still. So we do see the differential expression of the purple gene is higher in sample 2, but now, for example, a number of genes in sample 1 now look overexpressed compared to sample 2, when we know the ground truth was that they were equivalently expressed. And so in this case, just a simple normalization like the scaling isn't able to accommodate this differentially expressed gene being present in the sample. Example C, if you want to guess, it's going to have a very similar issue in that We're not going to change the zeros because we're just scaling by a multiplicative factor. So zeros will stay 0. And so these counts are still in the right direction for the ground truth. But now with sample 1 versus 2, instead of being, for example, instead of blue being equal across sample 1 and 2, we see sample 2 has higher expression still. So even though we've normalized using this approach, there's still this artifact left over. So in summary, counts per million can be dangerous when comparing across samples. It forces all samples to have an equal number of total reads, which is a very strict rule. And so basically that's assuming that each sample is going to have the same amount of expression within the same set of genes. And this is a known problem going back to even bulk RNA-seq. People were aware of this. And so there was more robust scaling factor estimations that were developed. So Andrews and Huber in 2010 introduced this median ratio method, and it tries to estimate a more robust scaling factor. So I'm not going to get into the the mathematical details of what's happening here, but you can I'll show you in a couple slides that this is actually going to be, instead of just assuming that things have to be fixed, that there's allowed to be some variability. So instead of just applying that equation directly, let's think intuitively about what this is actually doing, because there's other methods that are slightly different mathematically, but are doing very similar things. So if we're trying to estimate a more robust scale factor, however we mathematically do that, let's pretend for now that we knew that this purple gene was differentially expressed. And so let's ignore it and then recalculate the totals. And so we can somehow ignore it from our calculation of total sequencing depth and perform kind of this adjusted scaling normalization. Now we don't have the totals fixed, which is okay, because maybe in practice in real life, that's not a good assumption anyway. But now we can actually see that we do have roughly equivalent expression for the blue and the red gene here, similar to what the underlying truth is. And of course, we don't fix the zeros here, but they are still very comparable. And so in this case, this allowed us to reduce the bias of just the CPM type normalization. And then let's say again, we have our different shapes. Oops. 1 second. Okay, I think I accidentally replaced this table. My bad. I don't know what happened to my actual table here. I'll figure that out later, but let me just skip to the next example because they're all very similar. Let's pretend we can ignore these genes that are differentially expressed in the sense that they're not present in the second sample, recalculate our totals, perform the scaling normalization. And in this case, again, we do have this equivalent expression between the blocks in sample 1 and sample 2. So these approaches are more robust in that they try to estimate that scale factor, ignoring things like differentially expressed genes, either in terms of overexpression or just differences in the library content. And these are typically called distributional normalizations because they assume that on average, like the distributions are similar across samples, but that doesn't mean that the count total has to be the same. And it does actually successfully mitigate the biases due to differentially expressed genes, as long as they're less than 50%. So that median and that calculation does make the assumption that as long as less than 50% of things are not differentially expressed, it's going to work pretty well. Once you go over this threshold, there's not really good computational methods after the fact to handle that. You usually will need to resort to like housekeeping or spike in genes that are added in known quantities to try to recover an unbiased comparison. Okay, does anyone have questions at this point, or is anyone lost or kind of following along? Okay. All right, so far we've established that some samples or cells are sequenced more than others. And again, this is often out of our direct control. We could do downsampling to mitigate this, but that would result in a substantial loss of data. So I will say I've seen that approach done. I just don't recommend it because sometimes in single cell, like the variation and total depth can range from 10 to 100,000, and then having to downsample to 10,000 is, the experiments are expensive. We don't want to waste data. And so we've established that the underlying expectation then is that if we were to take a sample and sequence it twice as much, it should on average have twice as high expression for every gene. And so here I'm trying to establish that we're going to see that this relationship holds in traditional RNA sequencing. And once we move to single cell, we see increased variability in noise. And that's going to actually establish why the newer methods have been developed and kind of, why they've come so far from what we had in bulk. So just as an overview, imagine that I have, this is about 25 different samples, and this is bulk data. And so the x-axis here is log of the sequencing depth, which is just the total number of counts in my matrix or, in one column. So if I look at different genes, this x-axis will remain fixed. And then the y-axis is the log expression of this particular gene. And so you can kind of see an increasing relationship, but let's just draw a line so that it's very obvious. So we sequence more in general, we see more expression for the gene. It's a natural assumption. If we want to normalize the data, we should want to remove this relationship. So we know that this is just technical, just due to things being sampled more or less. And so if we remove this count depth relationship, then we have a successful normalization. And so we look on bulk RNA-seq across more than just one gene. So for just illustrative purposes, a really highly expressed gene in red, moderate and black here, and then in blue is a lowly expressed gene. We see that these lines are parallel. So this trend is holding of this proportional increase in sequencing proportionally increases the amount of counts that we capture for each gene. And if we fit a slope across all genes, all 20,000 genes in the data set, and then block them into groups based on their high or low expression, almost all the genes have this slope right around 1. So it is a very proportional increase. There's some 0 here, and that's mostly due to this is a quantile regression line, and when we get a lot of ties, the line will be flat just because of the fitting through the median quantile. And so if we apply a global scale factor method, you can see now the slopes are right around 0, pretty much for all the genes in the data set. And so we've removed this count depth relationship. And so the normalization is successful, and you don't have this technical artifact in the data. And then we move to single cell. And so looking similarly at a highly expressed, a moderate, and a lowly expressed gene, we see that there is increasing right in the high and moderate, but it looks like the highly expressed gene actually is higher than 1. So it's for some reason, we're sequencing that cell more, but getting more counts of that particular gene. And then for the lowly expressed gene, it's, we're sequencing more, but we're not getting more counts for that gene. And you can think of it as, I mentioned with the Legos, assume that there's an infinite number, we could just keep sequencing forever. But in a single cell, when we're starting out with potentially very different count distributions for a highly expressed gene versus a low expressed gene, and then we do a lot of PCR amplification, you can imagine in the sample that goes to be sequenced, that highly expressed gene is now way overrepresented. And then for the low expressed gene, we keep sampling, but the chances of seeing it are not changing, essentially. And so we see this variability here in what we call the count depth relationship. And so you can imagine applying a global scale factor isn't going to do very well. It's going to treat all the genes the same. And so we're going to not normalize the highly expressed genes very well, and then essentially over normalize the really lowly expressed genes. And so this really established that there's variability in the depth relationship for genes in single cell, and it also affects the variance that we're seeing in terms of the count variance of the gene. And so I'm not going to go into the details, but we did develop SC-norm, a normalization method for single cell data. It was published in 2017, available on Bioconductor. And I still get a number of emails of people using it. But I will say that it was developed with the full transcript length protocols at the time. So for 10x data where 80% or even 90% of your data are zeros, I wouldn't recommend using this method. There's newer methods that are going to tend to perform more optimally. And so we're going to take a look at those. Okay, so now we've established a goal of normalization. And so our basic goal is that we want to compare genes across cells accurately without a technical bias. And by far, the variation in the sequencing depth is the largest technical bias that we observe. So what's the, why are biologists having, like, why are we having this talk? What's the motivating factor? So there's a lot of tools in single cell. As of 2023, pulled this a couple of days ago, there's about 1,500 methods. About 10% of that is normalization. So that's over, almost 150 different normalization methods. And what that means is, for people developing methods, it means we don't have a consensus. And so for users downstream, they maybe don't know where to start. There's 100 tools to choose from, and everyone says theirs is the best. That can't be true. And then also, because of that, a lot of people use these pipelines where maybe the pipeline doesn't really always give you an option to choose from 100 methods. You might have only one or two choices. And so we're going to look into just what are the newer methods that have been coming out, and what are their assumptions for single cell. Okay, so after SC-norm, there was this SC-transform paper by Hafmeister and Satij's group. If you're familiar with Satija, his group developed Seurat, which is probably used by everybody in single cell. It's a really nice comprehensive pipeline, really great for getting started, and analysis, and just kind of looking at what signals maybe are in your data. And so at some point in time, he came up with this idea of not only should we normalize in terms of correcting for depth, but we should probably stabilize the variance at the same time. So we want some type of method that can do both of these functionalities. And just as a point of comparison, previously when we use scaling methods, or for example, SC-norm, we just focused on removing that depth association. And then afterwards, you could do a variance stabilization in the form of just a log of the normalized count plus 1. And that's supposed to stabilize the variance for most downstream applications. And so the SC transform is essentially fitting a negative binomial regression, and then taking the residuals from that regression and saying this represents normalized data. So we've just normalized out the depth effect, but also the way that they estimate parameters by borrowing information across genes results in a variance stabilization. And then not too long after, around the same time, the Towns et al. Paper introduced this GLM-PCA. So it is focusing on dimensional reduction, but within the paper, they suggest that if you just really want to get a normalized count matrix, you can use their approximate multinomial deviance residuals of their model. So they sound same, right? They're fitting a model and then taking the residuals and saying that those have effectively normalized the data. All right. And so there are potential issues with this joint approach, right? So this Laus, Behrens, and Kovac paper from 2021, they point out that model specification is important, and how you estimate those parameters can have a large effect on how good the normalization is, and whether you've actually removed the depth effect, or you've possibly smoothed your variance, and so you can lead to biases. And I will say, their recommendation was to get the normalized matrix via this analytic Pearson residuals. SC Transform actually updated their algorithm to reflect the results of this paper. But if you used SC Transform very early on, even as a biologist, you kind of would see things that looked a little weird, like genes that are normally very highly expressed. They would disappear from analyses later on because their variance was shrunk so much that it kind of dropped out in further analyses. And so this paper showed that the original assumptions were problematic in certain scenarios, but SC Transform has been updated to account for this updated procedure. A more recent paper by Bouchani reiterates that there is bias in the joint methods for depth normalization. And so they actually propose that since everyone's just doing this CP 10,000, just dividing by the total depth and then multiply by 10,000, what can we do afterward that will stabilize the variance effectively and not introduce more bias? And so they do compare the SC transform and this Will Towns approach as well as the approach introduced here. And they end up coming up with a solution that's a little interesting, and I haven't had a lot of chance to explore it, but they recommend that instead of just trying to kind of normalize the totals, they're also going to normalize the gene sums. So if you think about a matrix, normally we're just worried about the column sums, but now they want to also normalize the row sums. And so they do this procedure where they do this proportional fitting to normalize both of those, then take log plus 1, and then do another proportional fitting. And so this paper is still in bioRxiv, so it's still under, I'm assuming it's still under review, but I think this has application potentially in like visualization of data and clustering, but for other approaches, it may not be as ideal, just because the assumption of fixing the gene sums may not always be appropriate. And so the most recent paper was this comparison of transformations for single cell RNA-seq data. This is by the Allman, Eltz, and Huber, which have done a lot of work on normalization. They actually just focus on transformations again, which is interesting. They say the depth adjustment, let's just perform a simple scaling. And then in terms of transformations, there's delta methods, which this is again just taking your matrix, dividing by some scale factor, adding 1, taking the log. And so if you've ever used Seurat or any other pipeline, this is probably what you're doing, where S here would be the, this scale factor to adjust to 10,000. The GLM residuals, which is, the methods produced by SC transform. And then there's other methods more recently that are these latent expression methods, which they assume that we're going to try to model the gene expression really well, and then get the predicted or posterior expression from that model. And so they kind of compare these different approaches. I'm not going to, this is a lot, so I'm just going to kind of summarize. They have 3 different evaluations: consistency, simulation, and downsampling. And so in terms of these evaluations, they have these groups of the delta method, transformations, GLM, latent expression, count-based adjustments, and then the negative control, which is just only doing the scaling or only doing the raw expression. A higher value on the x-axis here is better. And so obviously just using raw expression or just doing simple scaling tends to perform really poorly across all their evaluation metrics. Overall, they say the delta method type transformations tend to perform the best, even on this B category, which all methods look, to do almost equally well, but also on category C, the delta methods tend to perform the best. And so this is the method they actually end up recommending at the end of the paper. So even though I just told you all these other methods for normalization, if you've just been doing like a CPM or CP10K, adding 1 and taking the log, you're probably doing pretty good. I do want to point out one other method here. And so I'm a little biased because I'm a co-author on this paper, but it's by Jared Brown and Christina Konchorski's lab. And so it performs, I would say, like at least as well as most other methods and better than a lot. But what isn't really pointed out in the paper is actually the variability here of the performance data set to data set. So some of these delta-based methods have a higher mean, but you can see there's quite a bit of variability in their performance. Whereas this dyno method, and then again over here, the variability data set to data set is actually quite consistent. You can see that just high variability over here. And so I think if you're looking at a benchmarking and deciding what method to use, don't always just look at the mean. You should look at how consistent is it performing, right? Because your particular data set could represent the one over here, or it could represent the one at the very end. And that might matter in the decision that you make. Yes, thank you for the question. So this benchmark does include the updated assumptions. So here they have the Pearson, which is from the third paper that I mentioned, and then SC transform is the original version, and then the analytic Pearson is the updated or the suggested update. I should check if they're using the SC transform original version or the updated version. That's a good point. So if they use the updated version, it should perform similar to the analytic Pearson. So if you're assuming if you're using the most recent version, it should look similar to this row here. Okay. So this dyno method, it is on Bioconductor and published a couple years ago. And so it is worth looking at because it has different properties than the delta-based methods, which I have actually in 2 slides. Maybe I'll just skip ahead. So to point out the differences is that scaling methods actually don't affect zeros. So you can imagine if you're scaling and transforming counts, if you want the zeros to remain zeros, but the other counts to change, you have to be careful about how a scaling transformation might shift the zero distribution from the non-zero distribution and how that might affect later downstream analyses. Whereas like residual-based methods or latent expression, they're not going to preserve the original sparsity. So it is possible for a zero count to not be exactly zero in the normalized data matrix. And so that does make it so that the methods are much more computationally intensive. So you can imagine just that log of y over scaling plus 1, it is instantaneous calculation, whereas these other methods may take a few minutes or even 30 minutes because they're Bayesian or based on different types of distributions that need more time to calculate. But doing so is actually potentially an advantage if you're looking at, for example, visualization or clustering procedures where having too much sparsity can throw off different distributional assumptions. And so if you if they're balancing out that posterior a little bit better, it may perform more optimally in downstream analyses. So going back a slide, just reviewing our assumptions. So what I've kind of pointed out here is that original normalization methods, we just cared about sequencing depth, but obviously with the advent of single cell data, we see that there's variability, and it's not just this consistent effect anymore. They do seem to be intrinsically linked. The way I imagine this is if you were to take your single cell data and if you just sampled all the sequences a bit more or more consistently, you would naturally stabilize some of those variances and maybe wouldn't need as much of a transformation. And so it does make sense that a lot of approaches now are trying to do both of these kind of at the same time. And so I want to add, if you're doing this in practice, what do you take away from all this information? Because again, there's a lot of papers and there's multiple benchmark papers out there. I would say to be critical in your analysis. So if you're just using a pipeline, look at what is it doing for normalization. Don't just run normalized data next step, right? What is it assuming underneath? If you want to be even a little bit the next level would be to maybe try a couple different methods, right? You can just try the top methods recommended by the benchmarking papers, and then look downstream a couple steps and just see how do things look? Are there big differences? And then if there's not, right, you can just pick whichever one you prefer, whichever one is most simple. But if you do see some big differences, then at least you have this information to go back and say, okay, what were the assumptions of this method and this method? Maybe I can figure out why they're different, and maybe which one applies to your data better. And so just knowing what information out there allows you to be able to do this. And then there is this Scone paper, which was published a few years ago. And so the idea is basically we can combine, pre-processing steps or even just normalization on its own. And we have these different evaluation metrics, and we can run all the normalizations and then compare for each metric which method seems to be performing, the best or maybe, underperforming. And so this can also give you a sense for your particular data set, how a given method is comparing to others. Because I think if anything to take away, especially from, I go back to this slide, is that there's just a lot of variability in the data set to data set performance. And so in some cases, you don't know exactly where your data set is. And it's good to just, look at what else is available and how it may be affecting your data set. Okay, so I'm hoping we can have like a discussion of just what other people think, other opinions. I do want to say thank you for the invitation very much. It was kind of interesting thought to put all these together. And so if you ever want to reach me, you can email me. If you want to reach me more instantaneously, you can just send me a message on Twitter. And I have my GitHub account here. And these are all the students that are in my lab, PhD students in my lab right now. So I'll go ahead and stop sharing and look at the chat, and then we can have an open discussion or just ask questions. Okay. Yeah, I think that's a great question. Is imputation recommended for single cell? Or how much would I trust existing methodologies? I did try to not get too much into the weeds because it gets more complex when you think about imputation. I personally don't do imputation. I would prefer to use a method like Dino that's basing things on this posterior. Yeah, it's tricky. Biologists also don't like imputation a lot. So yeah, I tend to stay away from it.
Yeah, I think just to follow up on that, I think I asked that question, and I tried something called Magic, which was I think a couple of years ago. I think it came out the entire pipeline for it, and I saw significant differences between but before and after imputation. Like how does clustering remain a little bit the same, but when you're doing the markers, like the find markers and all of that kind of analysis, things started not to make sense. And as a biologist, I decided not to kind of touch the imputation side of it. So that's why I was just curious to see whether you recommend doing it or not.
Yeah, I think so. I would say to me, the biggest difference between just these normalization or stabilization transformations is that they're generally, they don't try to do too much. Imputation, I think a lot of the methods, they try to make things a little too perfect. And then that really has the downside that it's picking up on some signals that are there, and then kind of smoothing your data set too much, to maybe it may be smoothing things out that are interesting, or it could be almost that it's too perfect of a result that doesn't hold up later on. And so I think that's my concern, and I think a lot of people maybe
right.
share
Yeah,
that.
agree. Thank you.
All right, I see a question about normalization and integration. What are your thoughts on the best practices for normalizing data sets and batch removal at the same time? Do we normalize the samples individually and then batch correct the integrated data set? Yeah, I think that's a really good question. I don't know that that's a, I know that I have an answer for it. I think someone needs to do a benchmarking. Yeah, I think someone needs to benchmark that to find out, because they're I know SC4, which is integration, they claim that they don't need to do any prior normalization. But then, for example, the Surround pipeline, which does integration, they're performing their own normalization within that pipeline. So I think it'd be interesting to see how that could change things. There was a paper that compared integration methods. I'll try to send it to Shamini. I think it was by Hao Wu. And I think he said that the normalization, I'll have to go back and look, but I think they did have some conclusions on how integrations and normalizations tended to interface, and his result was there's not a lot of effect. There's other biases that are kind of more of concern if you're worrying at that level.
Great. Thank you for your response. I had one other quick follow-up question. So if someone is using SC transform or other GLM-based normalization approaches, it would be possible to integrate batch as a covariate into the model, and other than depth, and kind of try to remove the batch based on that. I think our current understanding is that that's not sufficient for complete removal of the batch, but I was wondering if you have any thoughts on that maybe.
Yeah, I think that's a good question. There's so much there is a lot right now discussion, especially about batch effect correction, because a lot of methods that are doing this integration are essentially over integrating. They're removing real signals from the data, and I do think putting batch is definitely a way to maybe mitigate the potential to overcorrect. I think that could work. It'd be nice to see like that maybe done on like a simulated or like, you know, pseudo simulated data set. But I think that in theory, it sounds like it could potentially work.
Thank you.
Let me click the link this person posted about magic. Oh. Yeah, I agree. Yeah, I didn't want, I'm not going to be that controversial, but yeah, I would avoid imputation. Yeah. Are different normalization methods appropriate for different types? Yeah, so yeah, I wanted to mention that too. So a lot of methods, for example, for just like clustering and visualization, like it makes sense to give a normalized matrix directly for most methods. But then if you're going to do differential expression, you know, each method has its own assumptions of the distribution. And so if you've changed the distribution already, that's potentially problematic. And so for any differential expression tool that you're going to use, even for like trajectories, I would look into the method carefully and see if they have given a recommendation of, you know, oh, like we're estimating a scale factor in our model, and so you don't necessarily need to do anything, because yeah, it's going to be so method dependent on what they expect. Whereas in Seurat, their downstream testing is like a Wilcoxon or almost like a t-test. And so in that case, you'd want to give a normalized variance stabilized matrix, otherwise things will go really wrong. But methods that are already assuming negative binomial and have a scale factor as a parameter, it's better to give the raw data. Thank you for bringing that up. Any other questions? Does anyone disagree with anything? Just curious.
Yeah, feel free to give your opinion as well.
Oh yeah, so mass, yeah, that's a good point. So that method was actually developed around the same time as SC norm. It does something interesting in that it tests the non-zero distributions separately from the zero distribution. And I think people have moved away from doing that. I'm not going to say it's wrong or right either way. I think it's asking different questions. So for example, if you're just looking at comparing the zeros, you're kind of comparing like, you know, how often a gene is being expressed. Whereas if you think about the express portion, you're asking a different question. I think with 10x data, because someone posted that it's not zero inflated, whereas the previous full length data was zero inflated, and the fact that you would get things that were like all zero and then like a count distribution over here. In that case, it made more sense to do them maybe separately. Now with the 10x data that's not zero inflated, we're like getting more zeros is simply a sign that the underlying expression of that gene is low. Maybe mass wouldn't be the preferred approach in that case. Yeah.
Yeah. I've seen something similar to that, where like if I just use CRT's traditional Wilcoxon tests for fine markers approach, and if you just do all marker approach, and when you compare to that, when you use mass as the test method, for example, I've seen that you get almost 90% the same kind of the genes that are overexpressed in one cluster versus the other, for example. But then the log-to-fold changes are like vastly different. Like, with the regular like Wilcoxon test, I would just get literally 0.95 or like 1 at max. Whereas if I were to do the mass, it would go as high as like 3 or 4. And as I went into the methodology or like the math behind it, it looked like that it was doing its kind of own normalization to some extent, or scaling to some extent. I wasn't sure. So I used the actual like the RNA counts for these instead of like using SCT normalized counts. So I wasn't sure which approach would be better, like whether to use, the raw RNA counts and do masked, or use the SCT normalized counts and do CROT's fine markers approach, just to kind of see exactly what's coming up, what not. Because when I do look at the visualization of the, and just looking at the expression plots and so forth, they both look exactly the same. Obviously, basically they're just coming from the same data set, but the numbers, they're like drastically different. So I'm wondering which one would be the better way of looking at them.
Yeah, so I would also ask, like, if you're just looking at the difference in fold change as like the most striking difference, right? Are you if you're planning to use like a cutoff, is the ranking kind of similar? Because sometimes things are just scaled, but
Ranking is mostly similar. About maybe 10% of them, sometimes it disappears from, if I see something in CR's original, like a default approach, versus if I use the mass approach. But I do see, like, within top 5, I would see at least 7 to 8 different, same genes that would show up in the top 10 differentially expressed genes in the mass approach as well. It's just that the values, the differentially expressed, log to fold changes and the associated p-values will be a little bit different for those. Log to fold change, I've seen a lot more different than, so I just wanted to see, I've seen papers, people have been using one approach versus the other. I just wasn't sure, and the people tend, in single cells common, they tend not to kind of justify why they're using one approach versus the other. They just go with that and they just say it in the methods.
Yeah, I mean, and sometimes there's not a reason. I mean, they just did what was easier or what was like loaded on their on their R session. It would be nice if we had like a supplemental document of like, why did we do this? Not just what we did.
Yeah. But would it be, so which one would you recommend? For example, if we were looking at, absolute differences between 2 different clusters, I'll say you have 10 different clusters, but we wanted to kind of profile how these clusters are different from one another. In terms of the values of something that we can present to the clinicians and so forth. So they typically tend to go look at log to fold change, and how drastic that change is, and something that they will recommend so that we could do further testing in the clinical settings and so forth. But CROTs, it's a little bit, you get the similar trends, it's just it's more dampened down, whereas mass kind of gives you the values that actually kind of excites everyone else's eyes. So I'm just wondering, which one would you recommend?
Yeah, so I think it depends a little bit. I mean, I think it's good that if you look at both and you see pretty consistent rankings, that's a good sign. If there's genes that are dropping off, I would go and look at those genes, why might they be not showing up in this particular method, and look at maybe even just the count distribution for those genes, right? Are you seeing a lot of zeros and then a little bit of expression? Are you seeing the opposite? Are you seeing really wide, biloadal type distribution? I would look at those. I think for marker genes especially, the assumptions are a little different than differential expression of a gene across treatment conditions, right? Because for a marker gene, you want it to be not just highly expressed, but very specific to a cluster. And so in that case, it may make sense to think about, for the mass testing, if it's kind of focusing on the 2 parts separately, it could be, there's maybe a potential advantage if you're looking for something that has really high expression, that's very specific to a cluster. But it sounds like the approaches are pretty similar, but it's definitely worth investigating, just looking a little bit deeper and seeing why that might be.
Yeah, look at it. So when I compared them, one of the things I realized that mass kind of gave me almost a similar data, just that they was very easy to plot and visualize at least. And at the same time, usually you never go with the just statistics. You also look at the visualization before you actually agree to that stats, whatever the numbers have been spitting out. So I agreed with that the mass, when you're looking at the cluster by cluster differences and so forth, it did a little bit of a better job. But I just was like you said, no one's actually done that kind of a direct benchmark comparison for this. So I just wasn't sure. So I thought maybe I'll ask. Thank you so much for actually, that helps, that you're leaning a little bit more towards that too.
Yeah, either approach is valid, right? You just have to make sure it applies to your data.
Yeah, thank you.
I have a question.
Oh, yeah.
So so I work on tumor samples, and then often I have a lot of issues when I want to integrate them, and I usually leave them separate, but I don't have any issues when it comes to, let's say, a normal healthy brain from a fetus or an adult. Like, but is it, is a bit of a naive question. Is it okay to use different normalization methods on, let's say, different types of data because
Yeah.
you find that it just fits? And is it okay to compare their expression levels downstream?
Oh, okay. Yeah. So two questions. So yeah, I definitely think, yeah, things can be very data set dependent. And so for example, that scone method I recommended can really show like which normalization method has maybe a better performance. And there's different reasons they could be performing well, right? So if normalization is relying on not just like how variable the biology is, right? So maybe the tumor samples are much more heterogeneous, you know, or the patients are really heterogeneous, that could change the distributional assumptions a little bit. Also in terms of like the actual experiment, you know, sometimes experiments go better, sometimes not as good. And so just how much variability you're seeing in that, you know, that depth of relationship, it could also be in play. In terms of comparing them after the fact, I would not do that directly. There's other ways you could do it. It depends on your question. But for example, maybe looking at like rankings, or like summaries of downstream analyses, so like comparing the differentially expressed genes. But yeah, I wouldn't compare the actual values themselves just because they wouldn't really be comparable.
Okay, and then just one last question. From that benchmarking paper, the variance for each method that they had, did they have any sort of like a metadata that shows what kind of data sets perhaps
Yeah,
do better
yeah,
and
that's a great question.
which ones
So
won't?
I can also, I'm gonna send a copy of my slides,
Okay,
copy this paper. They actually have like a web interface, like Shiny app type thing that shows like all of the results and then also breaks it down by the data set that they used. So if you're like, oh, let me see if any of these data sets compare to mine, then that could be really useful. So yeah, they have a link in the paper for that resource, which is really nice.
okay,
Uh,
thank you.
And if I can ask, like a piggyback on one of your questions, Shamini's first question, where you mentioned that when you have normal data sets and when you have tumor data sets, I've been looking at the same thing. Then when I did not integrate my normal data set, though this was developing brain samples, and because biology made sense when I was looking at 5 different, let's say, time points of development, and everything made sense, like what I was expecting to see versus what not. But the tumor, when I did that, like there was just so much patient specific patterns that made me actually want to say that maybe I may need to use something like a harmony to kind of overlap them a little bit better so that I can actually see them. Would that approach still be okay? That for developing like brain tissues that I've been using, that I don't use any integration method, because at least biologically I'm not too concerned about it, because a lot of the things actually integrate. So like at least, or the clusters, they're not just like very sample specific. But when I do that with the tumor samples, it's always going to be like a sample specific clustering and everything, so that kind of gives you an indication that you need to kind of integrate to some extent. I do use harmony, but I relax some of the parameters so that way it doesn't force them so much, so I don't lose the biology too much.
Yeah,
Yeah, I wanted to
yeah,
kind of get your opinion on that. Yeah.
we recently ran into this. So I had a student integrating different patient samples, and harmony was kind of the preferred approach because it does let you kind of choose over like
Yeah,
which
control it a little bit better.
yeah, because it's really easy to see that depending on what factor you choose, you can get really different integrations. And so like I said, I think there's a lot more work coming out of about normalization or about integration very soon in terms of when to be very careful. But like you said, I think cancer is a really good example. I wonder if there's any like new literature like that's kind of targeted toward cancer in terms of integration, because it is such a big field and like particularly specific problem. But yeah, we chose Harmony and then looked at the different like integration factors that we could choose just to see which one tended to. I think there's something said for like, you know, looking at how much like consistency the integration has with like the factors that you expect should be overlapping or not overlapping. But yeah, I don't know of any good like evaluation techniques to know like which one is truly better besides just using your intuition and biological knowledge.
True. Yeah, I agree. Yeah, I think I'm not sure. I didn't do any benchmark study or anything like that. But originally, we had about 300,000 cells cohort, and when I tried to kind of integrate them using CROTs, like into like the default integration method, I realized that so every single clusters, everything was kind of it was perfect integration. But I realized like what I was losing, because like a perfect integration means that you're losing a lot of the differences as well. So you have to kind of keep it. So it's a trade-off. But when I did with Harmony, and I realized a lot of the things that were actually getting mixed out was actually the developmental. So that was one of the reasons. Like I decided to choose Harmony just because you can control the parent a little bit better with that. But I just wasn't sure. Like, is it still okay though? With the developing stuff, let's say if you have true normal, you don't integrate because let's say biology makes sense and everything, but you apply completely opposite approach with the tumor samples. Like, is it still? I mean, I think a lot of the time nowadays, I think biology will drive the kind of discoveries, but just want to make sure these numbers are also kind of backing that up as well.
Yeah, and like I said, I think yeah, with Harmony you can definitely do integration to say like do the integration like within normal and like within the tumor, and then there could be like some like semi cross alignment, but it's not going to force like normal to integrate directly with tumor.
Right.
That's what you want to avoid. Yeah.
Okay. Thank you.
All right. I think we have come to the end of our time.
Oh, great. Yeah, very nice. Thank you very much. It was a great discussion. It was great hearing you guys' questions and hearing that we share some of the same, roadblocks sometimes in analysis.
Thank you so much for coming and giving this is a very important topic. Yeah, and we'll definitely stay in touch through Panoramics.
Thank you for organizing. I'll definitely attend some future talks and learn a little bit more.
All right. Thank you, everyone. And thank you to our audience as well, who came down during lunch. So have a good weekend.